Good afternoon, fans of publicly supported science, and welcome to the BioEPIC Slam. I am Kat Adams from UC Berkeley and the MCAFE's SFA, or Science Focus Area, which you will hear more about later. And today we are coming to you live from Lawrence Berkeley National Laboratory, also known as Berkeley Lab, in the beautiful hills overlooking San Francisco Bay and the Golden Gate Bridge. This is where we bring science solutions to the world. We are gathered this afternoon to be regaled with concise science stories from early career researchers who will soon be housed in what will be the newest building at Berkeley Lab. To help me introduce these presenters, we have recruited talent emblematic of Berkeley Lab early career researchers. You know her as the champion of the 2021 Berkeley Lab Early Career Research Slam, and she went on to capture the crown of the inaugural Bay Area Research Slam, where she rose above a field of luminaries from Lawrence Livermore, Sandia, and SLAC National Laboratories. She charmed us all with a tale of microbes from fish guts that convert seaweed into sustainable biofuels. I present to you now from the Joint BioEnergy Institute, or JBay, the champion, Lydia Rachbauer.
Thanks, Kat. You're too kind. But by now we have a new champion, as Kat herself is the winner of last year's inaugural BioEPIC Slam with her amazing talk on, if you remember, funky fungal farts. Today, we are bringing you lightning presentations by some of the leaders of research now distributed across the vast Berkeley Lab campus that will soon be brought together in BioEPIC, the Biological and Environmental Program Integration Center, when the building is completed in 2024. Here in the virtual room today to tell you some more about BioEPIC are the two building leads, Susanna Trench and Peter Nico. Susanna is the Director of Environmental Genomics and Systems Biology Division in the Biosciences area, and Peter is the Deputy Director of the Energy Geosciences Division in the Earth and Environmental Sciences area. Susanna and Peter, please take it away.
All right, thanks so much, Kat and Lydia. It's exciting to be here, and we are here to celebrate BioEPIC, which you can see the diagram of in our background, but it's been amazing to see the progress on BioEPIC since we held this event last year. It was really just a skeleton a year ago, and now it's really transforming into a finished building. In EGSB, we are super excited to move into this amazing new space where we're going to have so many more opportunities to interact with each other and with other biosciences staff and with our colleagues in ESA. We are also excited about some of the unique capabilities that will be housed in and enabled by BioEPIC. These include instruments for cryo-electron microscopy, which is a powerful technology for studying the structure of biological systems. BioEPIC is also going to house the EcoBot, which is a prototype system for carrying out automated and even self-driving plant microbe experiments in small-scale fabricated ecosystems, or ecofabs. And it's also going to house a suite of ecopods, which are meter-scale fabricated ecosystems for studying plant microbe soil atmosphere systems under highly controlled and reproducible conditions. And then finally, designs have been completed for a greenhouse on the roof, which will be a unique and unprecedented resource for Berkeley Lab. So you're going to hear today about some of the incredible science projects that will take advantage of BioEPIC. And I think the synergies across these programs will really be evident. But before we get started on those presentations, I'll hand off to my ESA colleague and BioEPIC science co-lead, Peter Nico.
Thank you very much, Susanna. And as Susanna said, this has been a project and a vision and a passion of the Earth and Environmental Sciences area, as we call ESA, along with the Biosciences area. And these new capabilities that the building will have are really going across scales. And so Susanna mentioned the ecopods, and then our next step up, we'll talk about our smart soils testbed, which is a larger scale mesocosm experiment that can have heterogeneity of plants and water flow and lots of things like that we try to replicate in the field. And this will be a focal point for our EcoSense program, which is really about how do you develop better sensors to deploy both in experiments in the field and in the laboratory. And EcoSim, how do you take all of that data we're generating and put it into numerical models in order to represent the environment and to expand out into environments we want to predict what's going to happen into the future. And part of that is also connecting to the field. Susanna mentioned the self-driving aspect of the EcoBot. we're really thinking about self-driving field labs too, fields of sensors that will bring data into the building, use those models from EcoSim to predict what and when and where things should be measured next, and then send that request back out to the field to measure the right things at the right time. And we're all really excited for the building to come online in about a year. And with that, we'll stop and hand it back off. Thank you very much and welcome.
Thank you so much, Susanna and Peter, for introducing us to the BioEPIC facility. It sounds like a big deal, and we're looking forward to seeing more of it.
Yeah. So here is what you need to know about the BioEPIC Slam. You will hear from leaders of 4 science focus areas, or SFAs. Those leaders will introduce 2 researchers each who will seek to encapsulate complex science stories into a compressed 3-minute format in understandable language. But of course, you, the audience, will be the judge of that. You will each have an opportunity to cast your vote for your favorite BioEPIC Slammer. And through the magic of computing science, aka Zoom polls, we will identify the honorable mention, second runner-up, and champion of the BioEPIC Slam. We assure you it will be truly epic. BioEPIC, in fact. Furthermore, these presenters will not only be honored with your praise via comments in the chat, they will be awarded cool cash, and not crypto, the real US greenbacks. Regardless, all of these science storytellers you will soon hear are winners in their own right. Lydia?
Okay, let's get started with some trivia to warm up the room. So here's our first question. What years? Sorry. Oh, this poll is popping up. Too quick. So what years is the construction of BioEPIC going on to span from groundbreaking to opening its doors? Is it A, 2021 until 2024? Is it B, 2020 until 2023? So actually this year. Is it C, 2021, and will we have to wait until 2028 to open the doors? Or is it D, 2022, and we'll be opening doors in 2025? Or is it E, started in 2022 and opening up 2024? All right, so how are we doing on the poll? I think it looks pretty good. Poll's still open, I see. All right, so of course, you all got it right. The answer is 2021 till 2024, option A. You
Yeah.
just pay attention.
Well done, everybody. BioEPIC is about to open its doors next year. So Lydia, why don't you tell us a little bit about who's involved in BioEPIC?
Sure, let's meet the 4 BioEPIC anchor tenants whose leaders will introduce their science focus areas and our SLAM contestants. We start off with the estimable Margaret Torren, a woman known for literally getting her hands dirty. You see, Margaret is an internationally recognized expert on soil carbon cycling, seeking to understand the potential for positive feedbacks that amplify climate change and the potential for soils to act as a large carbon sink. Their team can often be found with their hands in the dirt, evaluating how soils respond to warming experiments at field sites from Point Reyes all the way up to Alaska. Aside from her professional pursuits, Margaret loves to visit the tide pools of West Marin, near where she grew up, and bakes, she might also break it, but she bakes a mean gluten-free chocolate chip cookie, I've heard. All the digging Margaret does help shine light on how climate change is influencing terrestrial landscapes. And so for our event today, Margaret is representing the below ground biogeochemistry SFA, which seeks to improve predictions of soil biogeochemistry as part of the soil plant climate system and the role of soils in global climate change. Margaret, let's dig in, shall we?
Yeah, thank you, Lydia. I'm going to try sharing my screen. Hope that works. Yes. So I wanted to start with another quick trivia question. And this is just, what is the most important greenhouse gas in our atmosphere? What is the one that most determines Earth's temperature? So we'll see if we get any chat answers for that. We may or may not. It's sort of an impromptu trivia. So we get a couple guesses here. CO2, maybe you're prompted by the picture. But actually, the correct answer is water. Water is the most important greenhouse gas, besides fungal farts. Thank you, Kat. Determining Earth's climate. But that was just for fun. I am going to talk about CO2. CO2 is the most important greenhouse gas that is directly affected by human activity. And in addition, on the natural side, the terrestrial carbon cycle helps maintain our climate by regulating atmospheric CO2 levels. So plants, as shown here, the plants take up CO2 in photosynthesis, and their litter is the organic input for soils. Microbes decompose this carbon, return it as CO2 back to the atmosphere. And this cycle is the product of many intricate interactions of plants, microbes, soil minerals, nutrients, climate. And microbes are really the engine. And this is one of the largest fluxes of CO2 to the atmosphere. So we need to know what happens when climate warms and stimulates these microbes. So the formal goal of the below ground biogeochemistry SFA is to understand the role of soil biogeochemistry in global change over the coming century. So we're trying to think big spatial scales and long time scales. Most carbon is stored deep in the profile. So to continue the puns, I'll say that we need to probe this question very deeply. And to do that, we use field experiments that warm the whole soil profile, and then we study a large suite of processes. And we're seeing that 4 degrees of warming, which is the amount of warming predicted by 2100, increases the flux of CO2 from soil to atmosphere by 30%. That's quite large. And to predict into the future, we're developing mechanistic models of terrestrial ecosystems that incorporate all this mechanistic knowledge. Now, I don't have I have the blue sky timer pinned, but I don't see a time, but I think I'll wrap up there and say that today we're very lucky to have 2 great slammers from our SFA. First, to start us off now, we have Ricardo Eloy Alves, a postdoctoral researcher who has been at the lab for almost 5 years, and his talk is titled, or his slam perhaps, "The Greatest Microbial Buffet on Earth and What Happens When the Food is Overcooked." I will stop sharing. And take it away, Ricardo.
All right. Everyone see my screen?
Looks good.
Yes, we can.
All right. Just, I don't see the timer.
If you go to gallery view, you should be able to see it, and then you can pin it.
Okay, give me a second. All right, I still don't see it. Okay, I think I see it now. Let me just pin it.
Let's restart the timer.
All right,
Give you
sorry
a
about
chance.
that.
You want to see Ricardo in the speaker view, right? Not the time. Right now, we're just seeing the timer. So maybe one of us can help let him know when the time is up or something.
Oh, okay. How do I now put my background back?
We see you with your background.
You do? Okay,
Yes.
perfect. Sorry about that. Sorry about the confusion. All right, so I'm about to start now. Can we reset the timer?
Yes, we will.
All right, so I'll just go. So good morning, everyone. And first, I'd like to ask you to try to picture all the trees and all the plants on the planet. And then try to picture all the gases on the Earth's atmosphere. So if you put those together, the amount of carbon that you end up with is just about only half of what all the carbon that you have in soils worldwide. And as it happens, these soils are also teeming with trillions and trillions of microbes that actually just, as Margaret said, they just love to eat up this carbon and just end up breathing it out as CO2 to the atmosphere. But actually, not all of these carbon in soils, which is mainly organic, in organic form, nor the microbial communities are actually made equal. So essentially, what you have thousands of different organic compounds in soils, in the same way that you have millions of different types of microbes, each of them with their own food preferences. So by analyzing the genomes of soil microbes, their protein expression, their activity in laboratory experiments, as well as the chemistry of the soil organic matter, we have actually seen that the most palatable foods for microbes, like sugars and amino acids, their concentrations decrease really strongly with soil depth. On the other hand, some of the foods that actually take a little bit more effort to chew on, like proteins and lipids, they tend to accumulate in deeper soils. On the other hand, you have other compounds that a lot of microbes also love, like alcohols, which appear to be very abundant throughout the whole soil profile. On the other hand, we also see that, for example, all this diversity of microbes also varies a lot with soil depth, meaning that also their functional capabilities also change, and food preferences, and as well as the type of metabolisms that are mostly active. They vary with soil depth as well. So what you end up with essentially is this gigantic microbial buffet under our feet, where not only just the type of dishes served, but also the food preference of the guests actually change very strongly with soil depth. Now, soils, as Margaret said, like pretty much the rest of our planet, are warming at incredible rates and worryingly rates. And as Margaret also mentioned, this is leading to increased CO2 emissions from soils, which is actually leading to a net loss of carbon, especially from the deeper soils. But how does this actually happen? Well, first of all, it's not so much that the food is getting heated up, but both the guests and the staff at the buffet is actually getting warmed up. So, for example, the enzymes that microbes send out into the soil to break down the big carbon chunks into small pieces that they can gobble up, they get faster with hotter temperatures. And you can think of this as like, say, over-caffeinated waiters, just serving all these small pieces of food to the microbes. And at the same time, we also see that microbial metabolism in general also becomes accelerated. So but all types of metabolism in general. So not only microbes actually eat more cake, but they also drink more booze. And what this seems to be leading to is actually that the soil carbon, the soil organic carbon, throughout the whole soil profile appears to becoming more oxidized. So not because more reduced organic compounds and bigger organic compounds, like, for example, lipids and proteins are being replaced by smaller compounds like carbohydrates, but actually because all of these different types of carbon are actually becoming more degraded and losing carbon. So what this essentially means is that, well, when the heating is on, pretty much every food at the buffet is fair game. So we are, right now, we are still putting together all these little pieces, but understanding these mechanisms will actually be a huge step forward in modeling and projecting what food from this buffet, how much of this buffet is actually ended up in the atmosphere as greenhouse gases.
I am very
Thank
sorry,
you.
Ricardo, but you're over time. I have to cut you
I'm
off.
done.
So you're done. Perfect.
Thank you,
Virtual
Ricardo.
applause for Ricardo.
Sorry, the timer was not preset, and I was always a bit confused.
Yeah, but now we're all hungry for lunch, thanks, and a good drink. So thank you. And next, I'm pleased to introduce Lucy Bennett. She's a UC Berkeley graduate student researcher, and she's been working at the lab or with our group for a year. And she'll talk about, can fun guys handle the heat? Mycorrhizal fungi and soil warming. Go, Lucy.
All right. Thank you for that introduction. And thank you also, Margaret, for introducing the soil warming experiment. I'm looking at our warming plots at Blodgett, and I'm exploring how mycorrhizal fungi will respond to soil warming, or can these fungi handle the heat? So For those who may not have had the amazing opportunity to know about these, mycorrhizal fungi are symbiotic with plants. They can expand the reach and surface area of their host's roots, which can help with water uptake and nutrient uptake. In exchange, they get sugars from the plant. So this diagram behind me represents that relationship. And I have a photo over here showing more what realistically the mycelium looks like. And so mycorrhizal fungi are ubiquitous in all terrestrial ecosystems. They associate with up to 90% of vascular plants. So they are everywhere around us. The fungus are among us. And so there are two main morphological types. There's endomycorrhizae and ectomycorrhizae. Endo reach inside of the cells of their host root, that's a root, and endo go between the cells of the root, but all mycelia go in the roots of the host. So my experiment is looking at two species of trees in our warming plots that one associates with endo, the other associates with ecto. And so in order to get to know these fungi more, I sieve out the roots from the soil of our soil core, pick them out, wash them, shake them, chop them, freeze them with liquid nitrogen, as you can see in these photos, and bead them, and then finally, they're ready for DNA extraction. And so what I'm trying to look for are, I'm going to be sequencing their DNA to get an idea of the total abundance of the different species, because I want to look at the community composition and diversity between our control plots and the +4 degree warming plots. And then I'm also going to collect some soil metrics and plant metrics to associate them with our community results. So why are mycorrhizal fungi important? We are interested in them because they are important for nutrient uptake, providing up to 90% of the nutrients. Okay, so how will warming impact the fungi? I realize I don't have a lot of time, but I want to explain that there's a lot of impacts that can directly affect these species, such as through increased plant growth and resource demands, or directly the temperature can change what species we're going to see because some species are more tolerant. And so when the fungal species composition changes, that can change what kind of seedlings can establish or what kind of benefits the trees receive from these plants. So that is why these relationships are really important to investigate further in warming. So thank you very much.
All right, there's our applause. Nice. Well done. Seriously, nicely done, Ricardo and Lucy. So are we ready for our next trivia question? Let's slam on ahead. So one of the 4 SFAs or science focus areas to be hosted by BioEPIC is MCAFEs, where I work. But what does MCAFEs stand for? Is it A, miniaturized computational assessment for ecosystem sites, B, microbial community analysis and functional evaluation in soils, C, multi-component assays for eukaryotic symbionts, D, man, complex acronyms freaking excite scientists, or E, massive combinatorial algorithms fostering ecology simulations? Make your guesses now. Where do I work?
People seem to know a lot.
Yeah, people are doing well. We do have at least one person voted for D, man, complex acronyms freaking excite scientists, which is delightful.
But of course, you knew it's B. It's microbial community analysis and functional evaluation in soil. So rolls right off the tongue. All right, to introduce AmcaPhage, we have Trent Northern. And among the many hats Trent wears, he serves as senior scientist and deputy director of the Environmental Genomics and Systems Biology Division. He is also the director of high throughput biochemistry of the Joint BioEnergy Institute, go JBay, and metabolomics program lead of the Joint Genome Institute. When he isn't immersed in leading multiple scientific initiatives, Trent has led multiple successful Bay Area food bank drives. And on top of that, he's also an avid winemaker and grower of orchids. Relevant to today's event, Trent serves as a laboratory research manager for AmcaPhage, where they are trying to understand and control microplant root interactions to support sustainable bioenergy. Take it away, Trent.
All right, thank you, Lydia. So I'm going to share my screen. All right, so it's my pleasure to introduce our program. You can see a picture of the scientist up here. It's a very diverse team, and we are focused on studying the rhizosphere. And so that's the region of soil immediately around the roots. And so you can see here a beautiful picture of plant roots covered in soils. And this is a hot spot for both carbon cycling and microbial activities. It's really important for the carbon cycle because, as you can see in this diagram here, carbon is being pumped into the soil by plants, which can release up to 20% of the carbon that they fix into the soils to feed beneficial microbes that provide the plant with important nutrients like nitrogen and phosphorus. So it's a really critical region to understand, both for the global carbon cycle and sustainable agriculture. But there are many challenges. There are complex microbial communities in the rhizosphere, and most of the microbes have not been cultivated. So we have very limited insights into their activities, such that most of the genes are uncharacterized, and many of the microbial interactions have never been characterized, especially the interactions between the microbes and their plant hosts. So AmcaPhage is developing a powerful new integrated approach where we perform in-depth genetic and biochemical characterization of the microbes within the rhizosphere. We construct spatial temporal models that allow us to predict the functions of genes and the interactions between the microbes. And then we use what we call in situ community editing, which is a powerful technology that you'll hear a little bit more about from Avery, where we're able to go in and remove microbes and genes from the rhizosphere and see how it impacts the system functions. And we do this inside of the laboratory in what we call fabricated ecosystems. And I have some pictures here for you of what these look like. So up here we see what we call an ecofab, and it's smaller than a shoebox, but it's a sterile system that allows us to study the interactions of plants and microbes under very controlled conditions. Here we have an image of a new ecopod that we've just gotten in the lab. We're really excited about this. It is huge. You could put a basketball hoop on there. It's over 14 feet tall. And this is another contained and controlled ecosystem we can use to study these interactions and use our community editing technologies. All of these are going into BioEPIC. So I want to quickly introduce our two speakers. Manuel Villalobos will be telling you about his studies of bacterial fungal interactions. He's been a postdoctoral researcher in the glass lab for 2.5 years. And he wants to know, in the rhizosphere, are symbiotic bacteria and fungi friends? And then Avery's going to tell you about CRISPR and anti-phage defense systems for doing targeted microbial community editing. And he's a 5th-year PhD student in the Baranju lab. So with that, I will pass it on to Manuel. Take it away, Manuel.
Hi, thank you, friend. So I am Manuel, and I like the fungal biology. And today I want to talk about the super complex mixture between bacteria and fungi that exists in the rhizosphere. some of the bacteria that are associated to the roots are good for the plants. And they, for example, give some nutrients as a nitrogen. But also exist some fungi that are able to promote the growth of the plants. And they are good for the plants. But we don't know exactly what happened when they are interacting, fungi and bacteria. But we know that trichoderma, that are a very nice fungi that you can see here of me, and is a fungi that produce so many compounds and improve the growth of the plants and protect the plants to the attack from other microbes in the soil. And some of these compounds are super complex and look like a weapons. Some of them are similar to the antibiotics. And it's super common because, you know, for example, penicillium, that is a fungi, produce the penicillin that attack bacteria. But that is happening in the soil. Maybe trichoderma is affecting the population of the bacteria and the bacteria are dying. We don't know, because we are thinking that maybe some of these good bacteria are collaborating with fungi. But to know that, we used a pool of mutants bacteria in so many genes, and we put this population of bacteria together with the compounds of the fungi. And we trying to understand what mechanisms are important to survive to these compounds in the bacteria, because we are thinking that the bacteria needs to support these compounds. So we found so very interesting results. For example, we know that all of the bacteria are able to survive with using many mechanisms, the antibiotic compounds, but also they can, some of the fixer nitrogen bacteria needs to take iron from the environment. And also they take purines from trichoderma. That is very cool because sounds like the fungi, the bacteria use these compounds, come from the fungi, and they are growing very well in presence of these compounds. Also maybe saving resources to biosynthesis of these compounds. So at the end, we are understanding that this is, as a whatever relationships, the bacteria needs to tolerate the presence of the fungi, but also take advantage and some good substance from the fungi to survive. That is very cool. Thank you.
Excellent. Thank you very much, Manuel. And next up from our SFA is Avery. Avery, take it away.
Cool. So hello, everyone. My name is Avery Roberts, and today I'm going to discuss our research on using genetically engineered viruses to make targeted changes to the DNA of bacteria within microbial communities. So our ultimate goal here is to apply these methods to the rhizosphere microbial community to better understand microbial interactions and how these communities actually impact plant health overall. So bacteriophages, or phages, are the viruses that infect and kill bacteria. And bacteria, in turn, can defend themselves from these phage infection events by using CRISPR-Cas systems, or CRISPR for short, which can locate and actually chop up the invading phage DNA to prevent the infection from happening. But since the discovery of this sort of mechanism, CRISPR has been repurposed as a powerful gene editing tool. And in our research, we're actually using CRISPR systems to engineer the DNA of phages that are infecting bacteria rather than having the bacteria actually destroy the phage DNA. So because phages have a real natural talent for getting the DNA into bacterial cells, we're using phages as a sort of delivery system to deliver actually CRISPR-Cas systems to edit the bacteria genome of the cells that are being invaded by these phages. So once the engineered phages delivers its DNA, the bacteria then produce the CRISPR machinery, which goes on to make precise changes or edits within the bacterial DNA. So currently we're engineering phages to deliver a specialized sort of CRISPR system that can actually insert huge chunks of DNA within the bacterial genome. And these insertion events are happening at locations of our choosing. So really anywhere within the genome that we want to insert something. And this technology will allow us to insert new genes or turn off genes within these infected bacteria. And we can even do things like incorporate reporter genes, so fluorescent genes like this GFP shown here, within the bacteria in their native community context. So with the successful development of these complex editing strategies, we'll soon be taking the next step towards actually editing members of the rhizosphere community. So a defined rhizosphere microbial community created by the MCAFE's team. And so this transition will allow us to actually use the advanced EcoPod growth chambers, which will be housed in the BioEPIC building. And they're designed for contained ecosystems and controlled environmental conditions. And the EcoPod provides a way for us to actually investigate how these plants and microbes react when we alter the DNA by delivering these engineered phages into these EcoPods with the plant and the soil conditions to our liking. So for example, in the EcoPod, we can use engineered phages to deliver CRISPR machinery that can knock out a critical gene and one of the microbes, and we can see how everything reacts in the system. So overall, we're developing flexible phage-based tools for targeted gene editing and microbial communities to deepen our understanding of how these microbes are interacting with one another, and overall, how those interactions are affecting the health and growth of the plant. Thanks.
All right. Well done, Manuel and Avery. That was seriously awesome. Okay, so who wants to play some more trivia? This next one's a doozy. So, which of the following is not one of BioEPIC's featured technologies? Is it A, EcoSim, B, EcoSense, C, EcoWare, D, EcoPod, or E, EcoFab? One of these ecos is not like the other. Which one is it? Cast your votes now.
I mean, we're not trying to trick you people. We can't, obviously.
They got this, Lydia.
Well, I mean, I don't even have to wait to end the poll. I mean, the answer is obviously and clearly, and I mean, everybody got that right. C. So that was not the hard one. But Kat, who's coming up next?
All right. It is now my pleasure to introduce Watershed function led by Owen Brody. Owen hails from Ireland and has made Berkeley Lab his home for a long half-life. He is Deputy Director of ESA's Climate and Ecosystem Sciences Division and serves as an Associate Adjunct Professor in the Department of Environmental Science and Policy and Management at UC Berkeley. And after a long day of advancing earth and environmental science, he likes to escape on his bike. The bigger the hill, the better, especially if there are cold beverages on the other side. We will now hear about the Watershed function SFA. Owen.
Super, thanks so much. Yeah, well, I'm delighted to be here to represent the amazing team that makes up the Watershed science focus area. And our goal is to build a mechanistic understanding of how mountainous watersheds retain released water, nutrients, carbon, and metals in response to disturbance. And our work is mostly focused in watersheds in the headwaters of the Colorado River in the Rocky Mountains. And you can see the East River right behind me here. In the East River and other locations nearby, we're trying to explore hydrobiogeochemical functions. So that's how water and elements interact and how microbes, plants, etc., influence the flow of water and nutrients and elements out of the watershed. And we work all the way from forests, as you can see here, down to the floodplains that receive the water and export it from these watersheds, and all the way from deep in the bedrock right up into the atmosphere. We follow the flux of water and elements. And so the approach we take, we're trying to divide the watershed into representative subsystems that are manageable to study and to explore how traits of the watershed interact to influence the function. So we're interested in microbes, minerals, plants, geology, topography, hydrology, etc., and how that all influences snow dynamics. And I wanted to focus on just one. I'm going to share my screen now. I wanted to focus on an aspect of this that is really about community building, because to do this and take something this complex on, it takes a village, and we take a community watershed approach. So we have a team of teams across multiple institutions, including Berkeley. We work with other networks, academic networks, studying these types of systems, and we work across interagencies as well to be able to leverage each other's unique skills and capabilities. We also develop and help create partnerships, both within DOE and outside of DOE with other national agencies. And we're working with minority serving institutions, et cetera, to try and build a better understanding and share our understanding in a more equitable way. And one thing I want to say, we're really excited about BioEPIC because one of the challenges we face is bridging all of these scales from microbes to watersheds. And the capabilities that are being developed in BioEPIC will allow us to come together with the new partnerships from biosciences and earth and environmental sciences to attempt to bridge these scales. So I'm delighted to introduce two of our SLAM representatives today, and both of these think deeply about the scaling challenges that I just mentioned here. So first up, we're going to hear about Mo Ombadi. Mo is going to tell us about flowing against the odds, the resilience of watersheds in the face of drought. And Mo has been a postdoc at the lab for 2 years. He's going to be starting his own group in the University of Michigan pretty soon. And he tells me he's probably the last person to have watched Game of Thrones. So that's his claim to fame. And take it away, Mo.
Thank you, Owen. The pictures on the left are or represent artistic depictions of two watersheds, the Truckee River mountainous watershed in the top and the highly urbanized San Joaquin River watershed in the bottom. While both watersheds have been subjected to prolonged drought conditions over the past 2 decades, the resistance of the two watersheds to drought is likely to be different. This is due to key differences in the processes governing the behavior of each watershed. For instance, water storage in snowpack in the Truckee River might enable the watershed to become more resistant to drought. On the other hand, reservoir storage and water demand play more of a dominant role in defining the resistance of the San Joaquin River watershed. As we try to think about the behavior of such watersheds in response to droughts, one of the key knowledge gaps is how can we define such an abstract concept of resistance? And this is exactly where my research comes in. A major theme of my research aims to develop statistical metrics using advanced data-driven methods to quantify the aggregated response of a watershed to drought. By developing such metrics, we are able to monitor the behavior of watersheds during droughts. For instance, the graph on the right shows the resistance of the San Joaquin River watershed during the California megadrought from 2012 to 2016. This figure clearly shows that the resistance started at a very high level and then decreased consistently and persistently as the drought progressed. Such an analysis enabled water resources managers to know exactly when to intervene in order to improve the resistance of the system. This can be done through regulation of water demand or controlling the release and storage of water in reservoirs. These statistical metrics also help us to examine complex hypotheses regarding natural and anthropogenic properties of watersheds and how they might interact with one another to define the resistance of a watershed. While my research specifically focuses on drought, some of my colleagues in the Watershed Function SFA project study other kinds of extreme events, including wildfires and heavy storms. They also use complementary approaches, such as process-based models and analysis of remotely sensed observations. Through our collective work, we hope to unravel the mysteries surrounding the behavior of watersheds in response to extreme events, and how that behavior might change in a warmer climate, thus making sure that these watersheds will continue to provide us with water, food, and energy for many generations to come. Thank you.
Fantastic, Mo. Thank you so much. Have we got a round of applause? Excellent. Yeah. Super. So now I'd like to introduce Helen Weyerbach. Helen is a data research scientist who's been at the lab for about 2.5 years. A fun fact about Helen, Helen used to help decorate the coconuts for the famous Zulu parade during Mardi Gras. I think we want to see some of that. So Helen's going to present about more than just water, how climate, plants, and microbes shape the future of watershed health. Helen, take it away.
Thanks so much, Owen. Mountainous watersheds, such as the East River watershed, which you get another view of behind me, are colloquially called the water towers of the Western US, providing drinking water to nearly 40 million people. In the last century, climate change has caused increased temperature and drought throughout these regions, which endangers the future of these water resources through increasing snowpack decline. But how do these changes impact the way that these watersheds cycle nutrients essential for plants microbes, and animals. Nitrogen is a key ecosystem nutrient, which is particularly limited in these pristine mountainous watersheds due to sparse vegetation and the strong influence of snowmelt-driven pulses of nitrogen, which are followed by periods of relatively few nitrogen inputs. Thus, in these areas of nitrogen limitation, nitrogen is a biologically important nutrient, which limits key ecosystem processes, such as plant growth and carbon accrual. Hydrology provides a key link to understand when nitrogen becomes available to the terrestrial system and how it moves to the subsurface and into the river, affecting other ecologically important metrics such as river water quality. For example, the residence time of water, or the time that it takes for water to travel through the biosphere, provides an important control on how long nitrogen is available to plants. Fast residence times, which occur during times of snowmelt, move soil nitrogen into rivers, streams, and into the groundwater, whereas slower residence time provides microbes the time to oxidize and reduce different nitrogen species. Thus, predicting changes in nitrogen cycling at the watershed scale requires precise hydrological process knowledge. To couple these processes, we've been working with our colleagues across the SFA to A, couple nitrogen processes with a high-resolution hydrological model, and B, develop relationships between water and nitrogen cycling based on field observations. When we scale these mechanistic relationships between hydrology and processes which govern nitrogen cycling using watershed traits, these traits represent bite-sized pieces of information that can have a large influence on the way our watershed cycles nitrogen, such as bedrock composition, land cover, and even the presence of cows. Using this model, we've run a series of atmospheric warming experiments to understand how these nitrogen dynamics could change in future decades. These experiments help us to start to investigate how future climate can impact the retention and release of nitrogen at the watershed scale. Not only does this conceptual model help us understand the future of watershed health in the East River, but it also gives us a methodology to apply fundamental knowledge gained from our watershed to other watersheds via watershed traits. Thus, these traits provide a key opportunity to understand how climate and hydrology influence the future of watershed health in some of our most vulnerable watersheds. It's more than just water. Thank you.
Super, thank you, Helen.
Thank you very much, Helen and Mo. I gotta say, today's presentations, all of them are absolutely top notch. So let's launch into trivia question number 4. What is the focus of the ENIGMA SFA? Is it A, figuring out where eels go to mate and lay eggs? Seriously, we still do not know. Is it B, uncovering the inner workings of plant roots and their effects on soil structure? Is it C, cracking World War II era encrypted codes? Or D, predicting impacts of microbial communities on ecosystems processes? Or last but not least, is it developing and solving world-class escape rooms? Some really good choices here for sure.
Yeah, well, do you think they'll get it, Lydia?
It's a hard one. It's really tough. I don't know.
Yeah.
I mean, I have my favorites for sure. I know what I would pick.
I mean, encrypted codes sound fantastic.
Mm-hmm. Who doesn't like a good escape room, though?
Also true.
I think we clearly have some favorites in the poll. And of course, the answer is D. Who got that one right? Everybody, well done, for sure.
All right, so now, but last, but certainly not least, we will get to know the ENIGMA SFA, brought to you by Adam Arkin. Adam has one proverbial foot on the hill at Berkeley Lab as a senior faculty scientist in the Environmental Genomics and Systems Biology Division. He is lead PI for the DOE Systems Biology Knowledge Base, or KBase, and scientific lead for the ENIGMA SFA. That other considerable footprint is planted securely on the UC Berkeley campus in his role as a professor in the Department of Bioengineering and lead for the Center for Utilization of Bioengineering in Space. He used to foster dogs before having to adopt the vicious and irredeemable Princess Peach. Adam?
Great, thanks, Kat. Well, ENIGMA is on the, let me share the screen here, one moment. There we go. ENIGMA works at a different scale, I think, than what you've seen so far. We are trying to go from the individual cells up to not even ecosystem scale, maybe just a few meters. But if you think about what our framing problems are, it's the one I'm showing you here in the upper left corner. You see, this is a map of the United States. And all those little dots are places where we have made some mistake or other in contaminating the soil with nitrate and uranium. This big thing you see in the middle is the Sagalaka Watershed, which is where we have pumped the water and over-fertilized to such a degree that the nitrates become very high in the water table, and uranium has been dissolved, and for the first time ever, Kansas farmers are pulling up uranium in their corn. And so we're interested in the processes by which microbes can reprocess that nitrogen, can immobilize that uranium, and ultimately sequester carbon. Now, we don't work at that little watershed over there. We work at a much more bizarre location. This is the Y12 site. It's a classified site at the Oak Ridge National Lab where we used to make nuclear weapons. And when we did that, we contaminated that watershed. And shown on this little diagram over here is this thing called the S3 pond, showing how small our scale is getting now. This S3 pond is where we stored a lot of that material, and then it cracked and it began to leak into the water table down this way. And now we are studying just this little area over here where the plume passes down into the water. And the idea is to build a completely mechanistic view, molecule per molecule, cell per cell, particle of soil per particle of soil, and so on, how the microbes process what's going on in this world. So to do that, we deploy lots of field operations where we can image this block of soil in this location over here to figure out what the soil looks like. We measure it in various ways using implanted wells or something we call the subsurface observatory, and we measure metals and metabolites and cells and microbes, and then we bring them back to lab and figure out what their genes and mags do. And to do all this stuff, it's a very large project with lots of cool people who span lots of different disciplines. And today, you will hear from two of them who are in the laboratory, taking that information we get from the field and then trying to understand mechanistically how the organisms that are achieving those functions work in the field. So to start out, we have Mingfei Chen, who is a geologist. He's been a postdoc with Romy Chakraborty for almost a year. He focuses on how microbes survive in the environments they live in, and in particular, how they make their houses, more or less, how they create biofilms that can survive on the soil. So he comes from a geology background. He's interested in using geological microbial record to better understand the Earth's history. And just as an interesting factoid, he started playing piano at age 6, and he still does it now. So Mingfei, take it away.
All right, thanks, Adam. Imagine a busy city below the Earth's surface, teeming with life and activity. But instead of skyscrapers and cars, the city is actually made up of tiny microorganisms, also known as subsurface microbes. However, unlike the surface environments, such as the soil, lake, and ocean, subsurface environments are usually more harsh, with no sunlight and limited nutrients. So it's actually very challenging for microbes to live there. But studies have been shown that the subsurface microbes might contain up to 70% of the Earth's total microbial biomass. So how can those tiny bugs survive and thrive in those harsh environments? One strategy is to form biofilm. You can think of the biofilms as the city's fortress. Just like the fortress that can provide shelter, protection, and resources to the inhabitants, the biofilms can actually serve as a safe haven for microorganisms to live in the harsh environments. The slimy matrix produced by the biofilms can act as a protective wall, shielding microorganisms from environmental stresses such as high pressure, low nutrient, as well as toxic chemicals. Additionally, microorganisms within the biofilms can work together for resources, just like how the inhabitants from the fortress can communicate and collaborate. Although biofilms are widely studied in the medical and industrial world, such as the oil pipe and oral biofilms, little is known about those environmental microbes, especially in the subsurface. So We believe that if microbes work together in the biofilm format, they might have a better chance to withstand those pressures and can even help to trap the migration of those contaminants such as uranium in the subsurface. In our research, we're looking at a certain group of microbes, genus Rhodanobacter, which are quite abundant in our highly contaminated field sites and are resilient to environmental stresses. When looking at how they can form biofilms that protect them from stresses such as toxic metals and low pH, we uncovered that certain metals, such as aluminum and manganese, actually can stimulate the formation of those slimy biofilms for those boxes that we have tested. We're now working with a group of researchers from the BioEPICs to uncover the secret genes behind the biofilm forming processes under those stress and use advanced microscopy to take an up-close look at how those slimy biofilm structures. Our next step is to put those bugs back in the field and observe how they behave in their natural habitats. Overall, in environments where life is tough and survival is key, biofilms show us how important it is to work together and adapt to the challenges we have. And that's all. Thank you.
Thanks, Mingfei. That was great. So presenting for us next is Bradley Biggs, who I have the honor of working with in my laboratory. Bradley has been at Berkeley Lab for about 1.5 years. And his research focuses on synthetic microbial communities, how microbes work together as a group. He comes from a chemical engineering background and is interested in how we would eventually apply what we learned to in ecology to engineering. So think about how we could use communities to do jobs you couldn't do otherwise. So Bradley enjoys being outdoors, but doesn't recommend overnight camping at Point Reyes in January if your sleeping bag isn't rated for it, because it is very cold. Bradley, take it away.
All right, thanks, Adam. So as Adam introduced earlier, our goal in ENIGMA is predictive microbial ecology. So what that means is we'd like to take things that are easy to measure in the environment, like DNA, and from that to predict the function of a microbial community. We want to know what the bacteria, the fungi, the archaea are doing and how they're interacting with each other. And eventually we'd like to be able to take these kind of measurements in any kind of environment, whether it's the carpet in your home or office, in the ocean or a wastewater facility. But right now we're focusing on this site in Oak Ridge, Tennessee, and the soil and subsurface environment there. And as Adam mentioned, this site has some historic contamination from some improper disposal of hazardous waste. So it has high nitrate concentration, heavy metals, and also it's quite acidic there. What's interesting is some microbes have not only survived this environment, but actually had the potential to remediate it. Now, for us to get all the way from measuring DNA to predicting a microbiome's function, there are a lot of gaps that need to be filled, and folks across the ENIGMA project are tackling different areas of this. Where my work comes in specifically is looking at how microbes interact with each other. And to do this, I create synthetic microbial communities, or SynComs, from microbes we take from the field, and we grow them together in the lab to see how they interact. From this, simple tests that are done in 96-volt plates, the strains are grown, and afterwards we measure how much of each one is there so we can know if the interaction was positive, if it was negative, or if it was neutral. We run this in many different conditions and can even mimic some of the hazards that we see at the site, like heavy metals and acids conditions. Now, from our tests, we've already begun to identify some strains that are both good and bad neighbors, meaning they're either cooperative or they're competitive. One example is Bacillus megatherium, which actually acts as the competitor to several strains you can see here. Another is Rhodanobacter fulvus, which is a good partner. Now, one thing you may notice is in both of these lists, the same acidivoric strain is there. So it's a bad partner with Bacillus, but the Rhodanobacter is a positive partner for it. In addition, all three of the strains shown here with the Rhodanobacter species were helped in the heavy metals and acids condition. So on their own, these three species actually can't grow in the presence of these harsh conditions. But with the Rhodanobacter around, they actually do. So the Rhodanobacter is not just a friendly neighbor, it's actually helping rescue them in this situation. So as we continue to collect data of these strain interactions, what we want to do is we want to use computational techniques to incorporate things like their metabolism, meaning what they eat and what they produce, so we can understand their interactions better. We'd also like to look for key features on their genomes that may be predictors of survival in a harsh environment, and maybe even help us predict successful new partnerships. Lastly, we'd like to take these interactions we study in the lab and integrate it with the measurements we take from the field so we can understand what we've observed there. Taken together, we hope to move closer and closer to predictive microbiome function and maybe even knowledge of how to engineer these microbial communities for other applications. All right, thank you.
Yay, Mingfei and Bradley. Thank you so much, Dankeschön, for the nice presentations.
Yes, so now the time has finally come, and it is your chance to vote for your favorites. I encourage all of the presenters to join us and turn on their cameras. And audience, first, let's show your appreciation and share your reactions and words of encouragement in the chat.
Yeah, give us some virtual love, please.
Yeah, thank you so much. Look at all that love. Thank you. All right. So you're about to see a link for voting, but just know, oh, there it is. The link is live. You'll have only 1 minute to cast your vote. The early bird gets the science worm. So to recap, we heard from ENIGMA, represented by Mingfei Chen and Bradley Biggs, the below ground biogeochemistry SFA with Ricardo Eloy Alves and Lucy Bennett, ENCAFE is represented by Jose Manuel Villalobos Escobedo and Avery Roberts. And last but not least, the Watershed function with Mo Ombadi and Helen Weyerbach.
All right, I think we only have 60 seconds for voting. Voting's open, so please be quick. The link's in the chat and make sure you vote for your favorites. So in the meantime, Kat, do you have like a bonus trivia on fungal farts maybe?
Yeah, I have a bonus trivia about fungi. So you all know that fungi are things like mushrooms and molds, but who are fungi most closely related to? Is it plants? Oh, we got a different one. Hold on. If you saw last year's BioEPIC Slam, you may recall my talk on funky fungal farts. What is the technical term for fungal farts? Is it gaseous metabolites, fungal pedita, volatile organic compounds, or pooty tooties? What is the answer? All right. I think we got it. Voting is closed.
Do they know? Do they know? I see the bars moving still.
Oh, bar's still moving. Bar's still moving.
We have a slight tendency, I see.
Oh, people are saying they can't vote.
Well, I think people are working on it behind the scenes already.
All right. Should we close bonus trivia and go back? Yeah, let's close bonus trivia and go back to maybe the bonus trivia got rid of the BioEPIC Slam trivia, or voting rather.
Let's see. So can you announce the correct answer?
Yes, the answer is volatile organic compounds.
Sweet. So people,
well done, everybody.
44% new. Organizers, can we get an update on voting? What's going on there?
Voting still
I know we were
closed.
using a third-party app for voting, and I don't know why it seemed that it was closed before it started. So we're looking into it right now.
All right.
Should be
Sorry,
working.
everyone. I know
People
we're past time. Hold
Yeah,
on.
thank you all for sticking around a couple minutes past time. I really appreciate it.
Yes.
These have been amazing talks. I really enjoyed each and every one of them, and I'm sure you're all struggling to pick a winner as soon as you're able to vote. And with this level of talent evident today, I, for one, am stoked to see how the BioEPIC facility will bring together under one roof our two areas' incredible staff and new capabilities, thereby further advancing an already close and fruitful collaboration between biosciences and earth and environmental sciences at Berkeley Lab.
Yeah, and many thanks to all the participants who represented our science focus areas today with their exciting presentations. After so many years of planning, it is fantastic to see the building rising up from the ground and to think we are so close to planning a move in. BioEPIC, as mentioned, will provide unique capabilities to understand microbe and plant function and in fabricated and natural environments even. So critical for success are, of course, the researchers themselves who will be performing their work in the building and linking that work to studies in the field. Today's presentations clearly showed that we are in safe hands, and we look forward to seeing many of you in the new building.
Yeah. All right,
So
voting.
I see that we're resolving the voting issue.
Yeah.
And voting should be working. Can we hear with a thumbs up, or can you show us with a thumbs up in the reactions if you are able to vote, people?
maybe
It
works
works now.
now.
Great.
It works.
Excellent.
Like it's
All
working.
right. So I will just verbally, while people are voting, do one last trivia. So you all know fungi are things like mushrooms and molds, but who are fungi most closely related to? Is it plants, slime molds, insects, or bacteria? And let me try to I can put the link in too. We can just put the link in many, many times.
There.
Who are fungi related to?
Oh, that's a tricky one.
It is a tricky one. It's not intuitive. I'll say that. So.
There's no clear favorite, I can tell.
No, it's evenly split, pretty evenly split. That's so interesting. Okay, I'll close it and I'll talk. So the answer is actually insects. So fungi are more closely related to animals than they are to anything else. And that's part of why it's so hard when people get like fungal infections. It's so hard to treat them because the fungus is so like at the DNA level related to us that it's hard to knock back the fungus without also hurting the human or the animal. So yeah,
That's
it's real
super
tricky.
interesting. I mean, the more we learn.
Yeah. And
That's so
slime
cool.
molds, oh man. So if anyone's seen The Last of Us, you'll see there's a slime mold at the beginning of the introduction to the show. It's not technically a fungus, it's a slime mold, but mycologists used to study slime molds because we thought slime molds were fungi. And then it turns out that slime molds are actually all over the phylogenetic tree, the family tree of organisms that have nuclei, eukaryotes. So.
Science is fascinating. It's crazy what we're doing and dealing with every day.
Mm-hmm.
So, okay, you slackers, I know we had trouble with the voting, but now you really only got 5 more seconds to log your vote. So let's do the countdown. It's 4, 3, 2, 1, and the voting should be closed now.
All right, now we're just using our special patented technology to tell us who the winners are.
Exactly. So there's a lot of math going on behind the scenes. And it was incredible and it was super fun actually to hear about all the research that will take place in BioEPIC and all the extra gross details about fungi, I feel, and the slime molds.
I'm just double checking something before we make our last announcement. Okay.
Yeah, we're waiting
so
for sure.
It looks like we've got 2 winners for honorable mention. We've got both Avery and Bradley, who will each get a spot award for $100. Thank you. Congratulations to them. Well done. Yeah, look at all that applause. Well done, everybody. And Lydia.
I think, okay, let's see. Do I see that? I'm not sure if I see the results, Kat. Please take it away if you can.
Sure, I can. All right.
Go ahead.
So our runner-up is Mo. Congratulations to Mo.
Applause.
Yeah, more applause. All right. So Mo will win a spot award for $175. Oh yeah, there's that applause. Excellent. Seriously, well done to them. And then last, but obviously not least, I get to announce the final, the next BioEPIC Research Slam Champion, who will get a spot award for $250 and bragging rights. And that goes to Mingfei Chen. Congratulations, Mingfei.
Congratulations. Kat and I both know how much work it is to prepare for such a slam. So you all did great. You're all our winners. And it's amazing that you're participating in such a great outreach activity. Let me say that.
Yes. And with that, I say thank you, Lydia. You will always be a champion in the pantheon of Berkeley Lab Slam champions.
Oh, thanks, Kat. It was a pleasure to host alongside the first ever BioEPIC Slam Champ. And let me say, the memory of your talk on funky fungal farts still floats on my mind. It positively lingers.
And thanks to all of you for tuning in today for the BioEPIC Slam. You can follow our upcoming events on social media at #BioEPIC. Be well and hope to see you soon. And stay epic. BioEPIC.
Thanks, everybody. Bye.